Greetings everyone. Today we're diving into Gene Set Enrichment Analysis tutorial in R. According to their website, Gene Set Enrichment Analysis is a computational method that determines whether a predefined set of genes shows statistically significant concordant differences between two biological states, such as different phenotypes. Without further ado, let's get started.
In step 1, we start by loading the required R libraries for our analysis. These libraries include HDO database, data table, fgsea, ggplot2, clusterProfiler, and enrichplot. Let's run our code. Next, we prepare our input data by reading in the results from DSEQ2 analysis. We extract the log2fold change values, remove any missing values, and sort the gene list in decreasing order, which is required for subsequent analysis. If you want to know how to extract the DSEQ2 results, you can watch our tutorial differential expression analysis using DSEQ2 in R. I will leave the link in the description of the video. So, to read the data, we use read.csv file, then we put the location of the file.
df equal to
Yes, here our data. So as you can see, the first column contains the gene samples. The second one is the base mean, then the log2fold change, standard error, statistics, p-value, and adjusted p-value. In this analysis, we are interested in the log2fold change column. So let's extract it with $sign. $sign, and then as you can see, we got the old columns in our data like X, base mean, log2fold change, and so on. We will enter for the log2fold change because this is a column that we are interested in. Then we name the vector names. We name here the gene symbol column, which is X. Then we remove the NA values or the missing values. Call it the missing values. Gene list dot vomit. Then we sort in descending order the gene list that we have created because it's required for clusterProfiler. So we're sorting sort yes decreasing actually. And now we set the organism and load annotation package. We specify the organism Homo sapiens and load the corresponding annotation package. This package provides the necessary gene annotation information for our analysis. So, set the organism Now let's run our code. Now let's perform Gene Set Enrichment Analysis. By using the fgsea package, we perform GSEA by providing our sorted gene list, specifying the gene ontology Go term as all. There is BP, which is biological processes, MF or molecular function, and CC, which is cell cycle. So we write all to gather all of them. We can also set other parameters such as number of permutations, p-value cutoff, and adjustment method. So let's write our code. We create a variable called GSE equal to GSE and then go, and then gene list equal to gene list, and ontology or ont equal to all. Key type equal to symbol, and n curve equal to 1000, minimum GS or gene set size 3 genes, and max gene set 100 genes. We put also the p-value cutoff to 0.05, to false equal to true, and org database equal to organism that we have created, key adjusted method equal to, we make it none. Now let's run our code. Now let's obtain a summary of our gene set enrichment analysis using the summary function. So summary, and we put GSE. So code, we got our summary. As you can see, we have the ontology, which is BP or biological processes, MF, molecular function, CC, cell cycle. In addition, we have the ID of the gene ontology. We also have the description of the gene set. For example, we have positive regulation of transitional initiation, regulation of insulin-like growth factor, peptidine, threonine, and so on. In addition, we have also the set size, which is the number of genes inside this gene set. So for example, here we have 12 genes, followed by 10 genes and 5 genes, and so on. Then we have the enrichment score and the normalized enrichment score, p-value, adjusted p-value, q-value, rank, leading edge. As we can see, the summary includes statistical measures and information about enriched gene sets, providing a comprehensive overview of the analysis outcomes. Then, to visualize our gene set enrichment analysis results, we will create the dot plot. So, dot plot show category equal to 8 split equal to dot sign. Here we adjust the angle of the x-axis labels, and then we adjust the font size of the y-axis label. Now let's run our code. We got our plot. As you can see, in the x-axis we have the gene ratio, and on the y-axis we have the nuclear membrane resampling, positive regulation of protein ubiquitination, cellular iron-iron homocytosis, and so on. In addition, we have two plots, one for the activated and one for the suppressed. The size of the circle is indicating the count size, from 5 to 15. In addition, the color is representing the p-adjusted value. Is the p-value high? Is it low? And so on. So for example, the highest number of p-adjusted value, which is blue color, is here. So the positive regulation of protein ubiquitination, this gene set has the highest number of p-adjusted value. And for the suppressed, here, this gene set, which is the positive regulation of transition and initiation, has the lowest p-adjusted value, which is the most significant, containing also count number, close to 10. We can also press here to make our chart looks bigger. So as we can see, the dot plot only displayed the most significant or selected enriched terms. While we may want to know which genes are involved in these significant terms, in order to consider the potentially biological complexities in which a gene may belong to multiple annotation categories and provide information of numeric changes if available. The gene concept plot or CNET plot was developed as a function to extract the complex association. The CNET plot depicts the linkages of genes and biological concepts, such as Go terms or kick pathways. So let's create the CNET plot. CNET plot, GSEA, let's run our code. As you can see, we have the glucose import gene set, which contains many genes, such as OCLN, DRD1, ITLN1, and so on. We also have another gene set, the phosphatidylinositol 4,5-biphosphate. This gene set contains two genes. The first one is INPP5D, and also SYNJ2. In addition, a third gene set, which is called the positive regulation of clathrin-dependent endocytosis, and it has one gene, DGKD. As we can see, if the gene is blue, it means that this gene has a negative fold change, but if the gene is red, then the gene has a positive fold change value. So as we can see, DGKD has a positive fold change, but for example, the PRKAG2 has a negative fold change, and so on. We don't only have the CNET, but the heatmap plot is similar to CNET. While displaying the relationship as a heatmap, the gene concept network or CNET may become too complicated if we want to show large number of significant terms. The heat plot can simplify the result and make it easier to understand. Let's make a heat map. P1 heat plot to GSE and show category 5. P2 heat plot also GSE fold change heat plot gene list. Now let's run our code. As you can see, we have a heat map contains of 5 categories. We have the regulation of insulin-like cross-factor receptor, signaling pathway, positive regulation of clathrin-dependent, phosphatidylinosytol for 5-biphosphate, nucleotide-activated protein kinase complex, and finally glucose import. We also produced another heat map to make it colored based on the fold change. So for example here, this gene, PRKAG2, has the most negative fold change, especially in the glucose import and in nucleotide-activated protein kinase complex, while it has a fold change value of 0 in the others. But for example, this gene, which is DGKT, has the highest fold change value, especially in the signaling pathway positive regulation of clathrin. Let's make it bigger from here. Now it's obvious and clear now. Let's close it. Now let's create a ridge plot. We will create a ridge plot to visualize the distribution of enrichment scores. Ridge plots are helpful to interpret up and down regulated pathways. So, let's run our code. Now let's make it bigger. As you can see, on the x-axis we have the enrichment distribution with values -10, 0, and 10. And on the y-axis we have the gene sets. For example, we have the activation of innate immune response, skeletal muscle satellite cell proliferation, skeletal muscle cell proliferation, cellular hyperosmotic response, glucose import, and many others. And here we can see the p-adjusted value. If the p-adjusted value is red color, that means it's highly significant. So for example, here in this plot, the highest p-adjusted value is belong to the glucose import, and the lowest p-adjusted value belong to the activation of innate immune response here. And this is also very blue, which is the protein kinase inhibitor activity, and so on. Now let's close it. Now let's create the GSEA plot to visualize the top enriched terms identified in our analysis. Let's run our code. And here we have it, our GSEA plot. As you can see, we have two plots. For the lower plot, the y-axis is for the running enrichment score, and it's from 0 to -0.6. And on the x-axis, we have from 0 to 25,000. And it's indicating for the position in the ranked list of genes. This point here is the enrichment score. The enrichment score reflects the degree of which the genes in a gene set are overrepresented at the top or bottom of the entire ranked list of genes. GSEA estimates the statistical significance of the enrichment score by a permutation test. To do this, GSEA creates a version of the dataset with phenotype labels randomly scrambled, produces the corresponding ranked list, and recomputes the enrichment score of the gene set for this permutated dataset. GSEA repeats this many times. The default one is 1000 and produces an empirical null distribution of enrichment scores. So, positive enrichment scores mean that the gene set at the top of the rank, whereas negative enrichment score, in our case, it means that the gene set at the bottom of the rank. Enrichment score must be adjusted to account for differences in the gene set sizes and in correlations between gene sets and the expression dataset. The resulting normalized enrichment scores allows us to compare the analysis results across gene sets. And what about this red line? From here to there, this area is calling a leading edge. The leading edge describes a subset of genes in a gene set. This subset contains the genes that appear in the ranked gene list between 0 and the point at which running some statistic reaches its greatest deviation from 0, which is the enrichment score. In our case, the enrichment score is about negative 0.7. The leading edge subset can be interpreted as a core group of genes that accounts for the gene set's enrichment signal, either positive or negative enrichment score. So they are the most interested genes. And for the second plot, which has the ranked list metric, as we can see, it contains many bars. Each single bar is a gene to give us an idea about the position of each gene in our ranked list. We can also generate another shape of the GSEA plot. Looks like the plot that we generate from GSEA software. Instead of GSEA plot, we have to write GSEA plot too. And here we have it. We got also the ranked list metric and the running enrichment score. And for the x-axis here, for the ranked list metric, we have the rank in order dataset, and is from 0 to 20,000 here and 25,000 here. If we look at the bottom portion of the plot, it shows the value of the ranking metric as we move down the list of the ranked genes. The ranking metric measures the gene's correlation with a phenotype. The value of the ranking metric goes from positive to negative as we move down the ranked list. Positive value indicates correlation with the first phenotype, or cancer in our case, and the negative value indicates correlation with the second phenotype, or normal condition. That's in case if we are comparing cancer to normal. And that's why here it's red color, indicating correlation with the first phenotype, or upregulation, and here a downregulation with the first phenotype, or correlation with the second phenotype. That's why here is blue. I'd like also to mention that the GSEA plot 2 also supports multiple gene sets to be displayed on the same figure. Now let's try to plot a GSEA plot 2 supporting multiple gene sets. GSEA plot 2, GSE, Gene set ID, we put from 1 to 3. It means that we want the first 3 gene sets. Well, we make each gene set with different color. So color, C, and we put the colors. Now let's run our code. As you can see, we have 3 different gene sets enrichment. The red one for the glucose import, the blue one for the positive regulation of clathrin-independent endocytosis, and the green one for phosphatidylcholine inositol for 5-biphosphate 5-phosphatase. And finally, let's create BMC plot. It's to visualize how unleashed terms have changed over time. PubMed trend of unleashed terms. And let's make it from 2010 until 2024. Proportion, make it code. Let's run our code. We did a mistake here. And here we have it, our plot. As you can see, we have 3 different curves. The red one for glucose import, the green one for phosphatidylcholine, and the blue one for the positive regulation of clathrin. The x-axis from 2010 until 2024. And the y-axis is indicating the number of articles. As we can see, for the glucose import, it has more than 8,000 articles, which has the most significant number of articles among these 3. So as we can see, this could generate the PMC plot showing the enrichment trends of selected terms over the years from 2010 to 2014. In the video description, I'll leave some links that aided me in creating this analysis. Please let me know if you are interested in learning about Gene Set Enrichment Analysis through GSEA software. Your feedback will help me decide whether to create a video tutorial on this topic. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel and hit the like button. Until next time, happy analyzing.